Alrighty, welcome to Chapter 19. This is where we will start Bio 202 for the fall 2020 semester. Normally we'd start with Chapter 18, but this time we're going to start with 19 so that we can kind of jump you in right straight into lab. Chapter 19 is focused on the blood, and blood is first before the cardiovascular system and before the lymphatic system, before the blood vasculature system, because blood is going to be all encompassing. So blood will be traveling throughout the cardiovascular system, and blood will contain the components for both chapter 18, the endocrine system, and for chapter 22, the lymphatic system. So we're going to start with blood here first. For all of my chapters, I always start with kind of a fun comic on the first page here on the left-hand side. So this is showing you a little comic about the platelet party. I think all of my pictures for the intro slide come from theawkwardyeti.com. The artist is fantastic. I think they're really funny, and the GIFs are awesome, and I definitely own quite a bit of them, so I'm a little bit of a fan. But I do think that the ones that I've picked out do a good job of kind of explaining or giving you kind of like a fun introduction to what we're really going to talk about today. Following the first slide, I always have a purple slide. The purple slide is going to give you kind of the objectives of what we're going to try to tackle for each individual chapter. So these are great for studying, but not so good for like first types of studying. So I would use these purple slides whenever you're going back through and making sure that you actually understand everything within the chapter. So let's go ahead and start with blood composition. Blood itself is actually considered a connective tissue. You might think, oh, it's just blood, whatever. You wouldn't consider it necessarily an actual tissue, but it's actually considered a type of tissue. And the functions of blood include transporting dissolved gases, nutrients, hormones, and metabolic wastes. So transporting dissolved gases is super important. We're going to see this whenever we talk about the respiratory system chapter in moving O2 and getting rid of CO2. Whenever we see nutrients, we're going to be moving nutrients in and out of the tissues, those that we need and those that we don't need. Hormones, whenever we're talking about chapter 18, whenever we're moving hormones from the place of where they were produced to the place of where their targets are actually at, so they can produce some sort of function. And metabolic waste, so as soon as the tissues are done doing whatever they need to do, they're going to produce some sort of waste, and that waste needs to be carried outside somehow, some way. So that will be carried throughout the blood. In addition, the blood is also responsible for regulating the pH. If you remember from 201, or from Bio 111, pH is arranged from 0 to 14, where 14 is really basic, and 0 and 1 is really acidic, with 7 being neutral. The P stands for a log-based score of whatever, log-based 10. And we use this to measure the acidity and basicity of any type of fluid within mammalian systems. So neutral for humans is anywhere between 3.45 and 7.35 and 7.45 is neutral. So the blood is going to help us regulate those extra hydrogen ions and bicarbonate ions moving back and forth, which we'll talk about. It'll also be important for regulating ion composition of interstitial fluids. So this includes like sodium, chloride, bicarbonate, potassium, and things like that. It's also responsible for restricting fluid loss at injury sites. So the blood will be responsible to carry platelets, and those platelets will be responsible for plugging holes that might occur anywhere within the blood or cardiovascular system so that blood doesn't leak out at any period of time. Blood is also responsible for defending against toxins and pathogens, and we'll talk a lot more about this in detail in chapter 22 whenever we talk about the lymphatic system and the immunity that we carry as humans. And last but not least, it's stabilizing blood body temperature as well. So heat is able to be generated by our muscular system. So as muscle is contracting and relaxing, we're creating a bunch of friction, and then all of that created heat from those muscle fibers contracting needs to go somewhere. And so in order for it to travel smoothly and easily, it'll actually travel through the blood and travel to the rest of the body. So this is why, like, whenever you're on a mountaintop and it's really, really cold, you're going to like do a couple jumping jacks or run up and down a little bit, or like start shivering, so that you create more and more friction and you create more and more contractions and relaxations of your muscle fibers, and then you can actually dissipate the heat towards the rest of your blood vessels, and your blood is actually going to help carry that heat throughout the rest of your body tissues. All right, so diving into 9.1 with the actual characteristics of blood and what blood is composed of. So first, the characteristics of blood. It usually is sitting around 38 degrees Celsius, which is about 104 degrees Fahrenheit, which, as you might notice, is a little bit warmer than what normal body temperature would be. And this makes sense because blood is going to be a lot more closely interrelated with our other tissues, like our muscular tissue, which is going to be producing a lot of heat. So it makes sense that the blood is actually going to be a little bit warmer than what our overall average body temperature would be. The blood isn't going to be immediately to the surface, and even if by the time it gets to the surface of like a person's skin tissue, it'll actually lose heat along the way. So it makes sense that it's going to be a little bit warmer than average body temperature. It's about 5 times as viscous as water. And what does viscosity mean? Viscosity just means kind of like the thickness, or like kind of like the gelatinous of a fluid. So kind of think the difference of pouring water down your drain versus throwing Jello down your drain, right? The Jello is going to take a little bit longer to actually be able to make its way through the drain. You might need a little bit more help to help dissolve it as it goes down the drain, versus water. Water is just going to pour right through. So the Jello would be more viscous than the water, and blood is going to be more viscous than water. The pH, like I mentioned, is going to be between 7.35 and 4.5 and 7.35 and 7.45, which makes it a little bit more alkaline, or a little bit more basic than what true neutral would be, which would be 7. About 7% of your body weight is actually the amount of your blood. So on average between men and women, it's about 5.25 liters, which is what this picture is actually depicting. Our 5-liter bottles full with, red food colored water, just to depict what all of the blood is that's ever circulating in your entire body at once, which I think is kind of cool, kind of creepy, but also kind of cool. All right, so blood composition, components of blood. So blood is really composed into 2 major parts, and so that's kind of what I want to start with our flow chart here. So blood, and it's composed into 2 parts. First is going to be the 55%, which is plasma, blood plasma. And blood plasma is essentially water, H2O, plus some proteins, relatively small, maybe medium-sized proteins, plus some nutrients, like some vitamins, and plus some hormones, which are some longer, more specified proteins. And then we have the second half, which is the 45%, and that 45% is going to consist of the formed elements. And the formed elements consist of 3 different parts. First are the white blood cells, and we'll talk a lot more about these in chapter 22. Those are going to be part of your lymphatic system. Then we have the red blood cells. And that's what's essentially, its essential role is carrying and distributing oxygen and getting rid of carbon dioxide. So here we're going to talk about erythropoiesis. So anytime you see erythro, that's referring to an erythrocyte, which is a red blood cell. And then every time you see poiesis, we're actually talking about creation of. So we're talking about the creation of red blood cells. And that will happen with the help of a couple of elements, which we'll talk about. But essentially, EPO is going to be our primary element here. And this will be released from the kidneys. And we'll talk about this again more in detail, just kind of introing it right now. We'll also talk about how they originate from a myeloid stem cell in the red bone marrow. And we'll talk about how those eventually turn into an erythroblast. An erythroblast, so anything ending in blast means it's predetermined to become something else. It hasn't quite formed its mature state yet. Then it'll turn into a reticulocyte. And then finally it'll turn into an erythrocyte, which is our red blood cell. Erythrocyte. And that's equivalent to a red blood cell. So those are both interchangeable. Make sure you're familiar with both of the terms. The third and last component of the formed elements are platelets. Platelets, as you'll learn in this course, are really important in clotting capabilities, but we'll actually learn that platelets are involved in many, many other functions as well. So essentially, formed elements you can equate to equaling hematocrit. And since many of you are interested in the healthcare field, I'm going to try to implement clinic-based language as much as possible in this course. So anytime you're measuring like hematocrit levels of a patient, you're actually measuring the formed elements of that patient. So if they have really increased white blood cell counts or really increased or decreased red blood cell counts, then you can kind of estimate what's actually going wrong or well with the patient at the time. Alrighty. Okay. At the end of some important units of the chapter, you can see that there are some relevant, and I'll actually zoom in here on the left-hand side, there are some relevant summary pages that will talk about the specifics of a certain portion of whatever it is that we talked about. So this is going to talk about a little bit more in detail what's involved in the plasma and what's actually involved in the formed elements. So definitely be sure that you're familiar with this slide. And then the picture up here, this flow chart, is exactly what we just drew out, except with a little bit more details. And we'll talk a little bit more about what the leukocytes are broken into. Those are our white blood cells whenever we get to chapter 22. Continuing on with blood composition, let's go ahead and deep dive into what the plasma actually contains itself. So the plasma is primarily water, like I mentioned, but it contains a lot of proteins. And those proteins are primarily produced by what organ, do you think? We have albumins, globulins, and fibrinogen. And so most of these proteins are actually produced by the liver. And this makes sense because the liver is constantly registering what blood needs to be recycled from the GI tract at any given time. And so whenever it's registering what blood has been used, it can actually also induce other pathways to make sure that we have enough of these proteins so that we can carry out additional processes throughout the rest of the body. Albumins we'll mention whenever we talk about transporting thyroid hormone. Globulins we don't talk about too much in detail. We actually just refer to them as antibodies, and we'll talk about antibodies a lot more in depth whenever we get to chapter 22. And then fibrinogen is what we'll talk about whenever we get to the clotting section of this chapter. The picture on the right just kind of shows you like a three-dimensional view of what's going on in a blood vessel and kind of what you would see if you were looking at the microscopic level of the different cells that would be within that blood vessel. All right, so the formed elements, so the plasma was 55%, the formed elements is the 45%. Formed elements we can consider hematocrit, so this is what can be measured from a blood draw. And in order to make formed elements, we go through a process called hemopoiesis. And hemopoiesis, remember that poiesis piece? That's creating of hemo. And anything referring to hemo means referring to the blood vascular system. So hemopoiesis of red blood cells is about 99% of what the components are of the formed elements. And then last, we have white blood cells and platelets. If we were to actually look at what this looked like in a test tube, so if we were to draw a vial of blood from a patient, for instance, and we were to spin it down so that the really heavy elements were on the bottom and the not so heavy elements would be at the top, we would see what we're looking at in the first picture here, where we see normal blood, and we would see the hematocrit on the bottom, and we would see the plasma on the top, and in between the two, we would actually see the buffy coat. Sometimes in pictures you'll see the buffy coat, sometimes in pictures you won't see the buffy coat. The buffy coat is actually just a separate portion of the plasma where it's just showing you those white blood cells. So they're just not as heavy as red blood cells, but they're not quite as light as the rest of the plasma elements, so they sit in the middle as the buffy coat, but you can consider the buffy coat as part of the plasma. If we were looking at someone who is anemic, losing the normal, or they had less than normal number of red blood cells, then we would see that their hematocrit would be much lower and their plasma would be much higher. And then the opposite would also be true for someone who would be polycythemia, where they would have an elevated hematocrit level, where they would have too many red blood cells compared to normal, and they would have limited plasma. All right, that's it for 9.1. I'll see you for 9.2.